So hello, my name is Eli Sword, and I'm a field application scientist at Geneyx. And so what I wanted to do is physically show our users in this tutorial how they can import a VCF sample. So once you log into the Geneyx analysis platform, you'll be presented with the dashboard view. The dashboard view is broken down into analyses and VCF samples. You'll see we provide you with really nice figures in terms of maybe cases that are opened as well as those that are unassigned, as well as a breakdown of all of the individual cases that you've imported into a particular analysis. So it'll show you modifications, analyses, number of samples, the status, as well as assignment. And you also have the option to go in and perform an analysis as well as searching capabilities. The VCF samples will show you the VCF samples that have been imported into the platform. Similarly, it's broken down into modifications, the VCF samples, the genome build, the version set, as well as if you wanted to start a new analysis, maybe with a different set of filter logic. You'll also notice on the left-hand side, we have a navigation pane. The analysis will break it down into the individual cases as well as the subjects, those reports. In the data management, we have the ability to perform processing tasks. So if you wanted to start from a FASTQ file, we do have that capability, as well as the options to upload single samples as well as those in batch. So maybe for high throughput environments. The settings options is a great interface, which we'll cover in later tutorials, but it gives you the options to make detailed kind of changes to your enrichment kits. You can enter in gene panels. You can modify thresholds for the ACMG classifier that is present in the Geneyx analysis platform. You can also modify protocols, filters, as well as regulate who has access to particular capabilities within the software of your team. But for this demonstration, I wanted to focus on creating a new analysis. And so you, in the dashboard view, can select the Start New Analysis option. What this will do is it'll bring you to the protocols. By default, we break it down into germline, somatic, and health screening. You can create a customized protocol, which will be displayed here, and you can select that for all future analyses. But for this example, what I'm going to do is select the trio analysis workflow. So the trio analysis workflow will bring you to the dialog of entering in the subject ID, which in this case is a demo trio. You can also enter in other relevant information, which can then be included in the final clinical report. Once you click on next, it'll ask you to define your VCF samples, which in this case, we have proband, mother, and father, and their associated VCF samples, as well as infection status. We do get questions if the Geneyx analysis platform can perform extended trio analysis, and the answer is yes. We can help you in creating that particular protocol. But in this case, we have all of our VCF samples associated, and if we click next, it'll bring us to the clinical information dialog. The clinical information dialog is rather important for entering in phenotypic terms. Once a phenotypic term is entered, it'll show you the specificity. So in this case, we have one intermediate and two highly specific phenotypic terms, which can be entered in through this dialog or the advanced option, or you can enter in HPO terms directly. Once you click on save, what it's going to do is it's going to annotate your VCF samples with gene tracks, frequency tracks, and clinical submission databases. The nice thing about the Geneyx analysis platform is it'll provide you with a notification directly to your email when that annotation process is complete, as well as provide you an audit trail within the Geneyx analysis platform. So in this dialog, we can see we have our analysis details. We can see it's the trio analysis. We can see the genetic models that were incorporated, as well as maybe if an individual user made a modification. So in terms of an audit trail, this is kind of a great resource. We can also see that the individual VCF samples have been annotated and are ready to be analyzed. So if we go back to our dashboard view and search for these particular samples, we can see that we have two samples that we can analyze. So if we click on the Analyze option, it'll bring you to the variant interpretation interface. So this is really a very intuitive feature, which we'll cover kind of in downstream tutorials, but basically each variant is listed on a per row basis. You can set up your filter logic however you see fit, and you can also see the different genetic models that are applied. Furthermore, we also have really nice reporting capabilities, but we'll focus this on different demonstrations. As mentioned, this particular tutorial is primarily for importing your VCF samples into the Geneyx analysis platform. If you have any questions about the workflow that we demonstrated or just questions in general, please reach out to support@geneyx.com. All right, thank you for watching.